In this episode of Voice of the Sea, we're analyzing data from the Exports Research Project. Exports is a joint investigation undertaken by NASA and the National Science Foundation to study the ocean's carbon cycle. During the summer of 2018, researchers headed out to the northeastern Pacific Ocean on board the research vessels Roger Revelle and Sally Ride. The goal was to collect data about water chemistry, currents, and organisms in order to better understand the ocean's food web and how carbon-containing material makes its way from the surface to the deep sea. Since returning from the expedition, export scientists have been identifying organisms, analyzing samples, and working together to make mathematical models of carbon flow in the ocean. We start off talking with Exports crew member and zooplankton expert, Dr. Debbie Steinberg, who is a professor at the Virginia Institute of Marine Science. Can you walk me through the process of studying the zooplankton?
To study zooplankton, the workhorse of the zooplankton ecologist is a zooplankton net. We have lots of different kinds of nets. We have fancy nets like our mock nests that we used on exports, which is a net that we can sample zooplankton in many different depth intervals all at once. And then we just have very simple nets that we just might want to just do a surface tow to get some live animals for an experiment. We also have some neat camera systems. We had a underwater vision profiler that's a video camera essentially that's lowered into the water, and it images zooplankton down through the water column.
Sort of in their environment while they're swimming around doing their thing?
Right. Yep. Once we get the tow comes back on board, we take the contents of the tow to the lab, and so processing the tow there'll be a certain amount of sorting and counting, the sieving, weighing and freezing animals.
Oh, I love them. Wow. Baby squids. Oh, I see.
And then another major part of what we do is live animal experiments. And so on exports, we were catching live animals. The live animal experiments are with these small zooplankton, and they might be done in a jar this big with seawater, and we might put 20 copepods in there to do the experiment. Or sometimes we'll use a larger animal, maybe a krill, which is a shrimp-like animal, and there might just be one of those in there. We were measuring how much they respired and also how much they pooped. And so all those processes that we care about, the role of zooplankton, the carbon cycle, we were trying to measure those on board the ship. So once we're back at our home institutions, a lot of the work actually happens there too, because much of the counting of organisms needs to be done in a lab that's not going like this, like it is on a ship. So it's much easier to do microscope work, for example, back in the lab at home. And so we did a lot of our counting then. There's a lot of data analysis. That's a big part of it.
What did I catch? Ooh,
Wow.
Teeth.
That's called a viperfish.
That's a huge one, too. That's twice the size of ours.
Those teeth are.
Look at those teeth.
Next, graduate student Connor Shea from the University of Hawaiʻi School of Ocean and Earth Science and Technology explains how he uses chemistry to determine what kind of food plankton are eating. What kinds of questions are you interested in looking at?
Where does the food come from? For a lot of different ecosystems, these dietary sources are really important. For instance, when you look at survival of different populations of salmon in the Pacific and the Atlantic, what their primary forage source is going to change their fitness and their ability to get home and reproduce. And you can use isotopes to track changes in dietary sources. And you can also use isotopes to learn a lot about animal movement. What my job looks like is I take this ground up plankton material, I do some chemistry, which basically consists of first extracting the amino acids. We have this raw biological material, and we need to pull these specific amino acids out of it because that's what we're interested in analyzing. And then I'll go ahead and purify those and get rid of sugars and polyamines and any sort of junk that we don't want in there to try and get it as pure as we can. And then we do what's called a derivatization, where we just change the chemistry of the amino acids just a little bit. We tweak it so that they play well in our instruments and our analysis goes a little bit more smoothly. And we're seeing that the base of the food web is composed of much smaller particles than we expected. These larger particles are generally thought to be kind of energy rich. They're sinking quickly, and so they haven't had a lot of time to be worked over by microbes. And so you assume that these are going to be the important dietary sources, especially for larger zooplankton. But what our analysis is showing us is that these really, really small particles, these particles that are sinking so slowly that they're almost suspended in the water column, are actually acting as a really, really important food source for the entire zooplankton community. It's like having a banquet of 100 people, and you're watching the food come in, and you just see one turkey show up on the table. And then everyone leaves the banquet hall like totally stuffed. And you're like, how did all these people get full on just one turkey, right? Sometimes we have this problem where we don't see a lot of material coming into this community, but yet there's all this activity down there. So the question is, okay, where is this material coming from? These suspended particles, you can imagine, have probably been hanging around for a while. They're not sinking particularly fast. And so they've been worked over by microbes for a long time. And that is one of the things that our analyses tell us. When these microbes act upon the suspended particles, they change the chemistry of that. They change the isotopic ratios in these particles, so we can see that. And we see that the small particles are extremely enriched in nitrogen-15. What this tells us is that there's a lot of microbial reworking of these particles happening, much more so than the large particles. And so a consequence of that is that a lot of the material that would be easy to metabolize has probably been removed from these particles by the microbial community.
What did the other exports researchers think of your findings?
I was coming in telling this story. I was a little nervous because I was telling a story that basically says, like, we have these particle traps, and our data seems to indicate that those particle traps aren't accurately capturing this important component of the food web. So coming into it as like a young scientist who's new on the project, I'm kind of nervous to share this information. Like, who's this guy and where did he come from telling me that my particle traps are doing a bad job, you know? But actually, it's not incredibly new information. This is stuff that people have had suspicions of for a long time. I was expecting a lot of criticism, and the community was generally pretty receptive of the ideas.
The University of Hawaiʻi Sea Grant College program focused on Hawaii's coasts and its communities through sustainable development, safe seafood supply, sustainable coastal tourism, hazard resilience, and healthy coastal ecosystems. Hawaii's Sea Grant.
Welcome back to Voice of the Sea. We're at the University of Miami Rosenstiel School of Marine and Atmospheric Science with Dr. Hilary Close and her organic chemistry lab.
I'm an assistant professor here, and I have a geochemistry lab. We do basically organic chemistry. We have fume hoods and glassware and stuff like that, and then we have instrumentation where we analyze individual organic compounds and their isotope ratios. I can show you this now. I wanted to give you an idea of some of the concepts we're thinking about. So first of all, this is about 1 liter of seawater. I pulled this from right outside this building. We're right on Biscayne Bay here in Miami, Florida. This is particulate matter. Anything that you're seeing that's a solid, that's a particle, it has gravitational settling. It's going to settle through a water column. So that's why we're interested in it. Anything that has been produced by life that is a solid material is going to settle through the water column and potentially carry that carbon into the deep ocean. So we're very interested in knowing the details of how that happens and how much carbon manages to settle through the water column versus getting eaten up and destroyed by animals or by microbes. Ooh, I think there's actually some stuff alive in here. I think I just saw something swim by. That would be a zooplankton.
That's awesome.
So what we do is we pump water through a filter in order to capture that solid material on the filter. So this is 1 liter. We tend to collect hundreds to thousands of liters of water. When I say hundreds to thousands of liters of water, we're not capturing the water itself, we're filtering out the solids that are in it.
Can you tell me just a little bit about your involvement in the Exports project?
We are looking at the transfer of carbon from the upper ocean into the lower ocean. What we're really interested in from a climate perspective is how much carbon makes it to the deep ocean, but it has to make it through this gauntlet of microbes and zooplankton who are eating it. So we want to look for evidence of who is eating that carbon, what is happening to it during this pathway. We normally think about what do organisms need to live? They need carbon, they need nitrogen, they need phosphorus. Those are the big ones. When you learn in high school biology, it's like carbon, hydrogen, nitrogen, oxygen, phosphorus, sulfur. But it turns out that iron is super, super important for primary producers. And iron mostly comes from the continents, either from rivers or from dust, like from deserts getting swept up by the wind. And the Northeast Pacific is sort of a notorious location for having not great iron sources versus a high nutrient and high micronutrient area like the North Atlantic.
Next, we talk with Shannon Doherty about her role in tracing carbon through plankton poop.
I'm focused on the fecal pellets of the zooplankton, which it's a very charismatic topic to talk about because I can summarize my dissertation topic. It's like, I study plankton poop. That's what I study. The zooplankton, they both eat and produce organic particles. So there's this whole mechanism by which zooplankton take in particulate organic matter and then produce it again. And when they digest it, it changes it chemically. So it's part of the particulate organic cycling in the ocean. Zooplankton are kind of the mechanism.
And so how are you studying what zooplankton do to their food and how they digest it and poop it out?
We have the three pieces of the puzzle, so the particles themselves, and then we have zooplankton bodies, so their biomass. And exports, the 2 zooplankton that we have that were really abundant there were salps, which are these like bags of jelly basically with an intestinal system, and amphipods, which are these little crustacean-y, they look like baby shrimp. And they are really interesting because they live inside the salps, so they're symbionts with the salps. They're eating what the salps produce and eating the salps themselves, which is really interesting. So we have those 2 zooplankton biomass, and then we also have the fecal pellets from those 2 zooplankton too. So the amphipods were separated from the salps. We collected their fecal pellets, and the salps collected their fecal pellets. So we have particles, zooplankton, and zooplankton poop. So we have the three things, and we can look at the chemical composition and the trophic dynamics of each of those individual things and kind of piece together what the zooplankton are doing to change the particles.
And what are you finding out so far?
In the exports region, the salps seem to be eating a lot of particles and phytoplankton, but not actually digesting them very much. So they would just eliminate intact phytoplankton. The way that we know that is we look at the trophic position of these animals. So that's the place that they are in the food web. And we can do the same thing with the fecal pellets. And then fecal pellets should look like both the biomass of the zooplankton and the diet, because there's some digestive workings going on to transform the diet, but there will be some things that are undigested. And so when we look at the fecal pellets, we want to see something in between what we would expect the diet to be, what trophic level we would expect the diet to be, and what trophic level we know the zooplankton is. And in the salps, their fecal pellets are essentially the same as their diet, which is telling us that they're probably not doing a lot of digestive alterations chemically, so they're just kind of pushing something through. Which is interesting because if that's the case, then they're this really interesting vector for taking in particles, and they're not really chemically altering them, but they're packaging them really tight in fecal pellets.
Next, Paul Vithol of the Close Lab in Miami shares some of the complexities of dealing with lots of samples and lots of data.
I've been mostly working on sample processing and sample analysis, as well as working on data analysis. Everything that has lived and will live on Earth probably depends on proteins and amino acids, and so trying to understand how those change is what we're trying to look at. My background is in chemistry. I like to understand these types of processes. Hilary and I just had a meeting. We were just talking about the fact that we have way more samples than I alone, or she and I, or we have 2 other grad students and our lab technician. We have more samples than all of us could actually analyze. Manpower is really another limiting factor in our analyses.
We're looking for a few heroes, mentors, trailblazers, innovators, a passion to change lives, spark curiosity, open hearts, and awaken minds, help students answer the question, who am I? This could be your calling, but this is no job. It's the journey of a lifetime. Be a hero. Be a teacher.
Welcome back to Voice of the Sea. We're at the University of Hawaiʻi in the Biogeochemical Stable Isotope Facility with Dr. Brian Pope, learning about the role of carbon cycling in the ocean food web.
We found that below around 200 meters, most of the particles that form the base of the zooplankton food web are really tiny particles that have been altered by microbes, significantly altered by microbes. And it was a real surprise. It's like going to a luau. If you're first in line, you get a good choice of what to eat. But if you're last in line, maybe all that's left are the things that nobody else wanted to eat. And we're seeing that in the deep ocean. The stuff that's easily digestible seems to be eaten by zooplankton that are living shallower in the ocean. And those that are living really deep are relying on sort of the dregs. It's what's left over and what microbes have been consuming, partially consuming. So they tend to be altered, and they tend to be really, really tiny, on the order of 1 to 5 micron particles. We would like an understanding of the carbon cycle in the next 10, 20, 100 years, 75 years. So really that time scale. So sequestration of carbon in the deep ocean is significant on that time scale, because the circulation time of the ocean is on the order of many hundreds to 1,000 years. We're looking at the changes we can expect in the next 50 years. That's significant. And that's something we would like a better handle on, so we can urge people to make changes tomorrow, today.
So what you're saying is that understanding how much carbon the ocean can take up in, say, the next 20, 50, 100 years will inform our understanding of what's going to happen in terms of our atmospheric carbon dioxide and its effect on our global temperatures?
Yes, that's really mainly what we're after, because there's a lot more total carbon in the ocean than in the atmosphere. The atmosphere is a small portion of the carbon budget, but it's really, really important in terms of the temperatures on Earth because of its greenhouse gas effect.
Next, Dr. Munso Rokaya.
Munso Rokaya. Martí, yeah, without a final N. Martí, yeah.
At the Woods Hole Oceanographic Institution in Massachusetts discusses her role in using naturally occurring radioactivity to trace the flow of carbon, and how she is working to combine her data with other researchers in the Exports project.
I'm a postdoctoral investigator at the Woods Hole Oceanographic Institution. I am part of the Café Thorium team, and we are interested in these radioactive elements such as thorium-234, polonium-210, which gives us information about other elements and processes that we are interested in. For example, this sinking of particles in the water column that regulates the climate too. So that's a really big important question that we have. The data that we are obtaining, it's really powerful. We were at sea for long enough, 1 month, that we can see the evolution and we can disentangle the spatial variability from the temporal variability. That's what we are working on. This is not quite common, right? To have this opportunity to go at sea for such a long time, so many people being involved, different groups, looking at the same processes, but from different perspectives, the chemists, the physicists, the biologists. So that's a really big opportunity. I was very excited to be part of this team and also learning from each other's work and seeing all this new technology that we deployed.
Finally, Exports lead researcher Dr. David Siegel at the University of California, Santa Barbara shows us what to expect in the upcoming Atlantic leg of the Exports research project.
What we saw was a situation that was much more animal mediated. In the Atlantic, we think because there's such strong vertical currents that can drive small-scale blooming events, that we do expect to see more of the nutrients driving phytoplankton growth, driving excess production that's just got to go someplace where it makes sinking aggregates that sink. The North Pacific, was supposed to be boring. The currents were teeny-weeny. The fluxes were going to be pretty small. I mean, we all knew that coming in. And we found that in the first part of the experiment, it was driven by salps. The second part of the experiment was not. The third part, salps came back, but the total fluxes were indistinguishable across those periods. So we have a largely animal-mediated flux production system that was invariant in time, which makes absolutely no sense. Half the time we didn't see them. They just disappeared. And how they live and all that sort of life cycle stuff, it makes it hard to be a modeler. When salps make their fecal particles, when they make their poops are huge. They filter huge amounts of seawater, and they have these feeding apparatuses that let them take very, very small phytoplankton and bacteria up to very large things. And they're able to eat anything. And they make poops that sink like rockets. They sink at 1,000 meters a day. It's quite remarkable.
Exports scientists will head to the Atlantic for comparison studies in spring of 2021. All Exports data will be made publicly available online. Follow us on social media at Voice of the Sea TV. Learn more and watch episodes online at voiceofthesea.org. Mahalo for watching Voice of the Sea.
Exploring Our Fluid Earth is the dynamic curriculum developed by the University of Hawaii's Curriculum Research and Development Group. The award-winning Fluid Earth and Living Ocean textbooks are now interactive and online. New activities, updated content, and a teacher community. Exploring Our Fluid Earth is now freely available. Find out more at exploringourfluidearth.org.